It's my pleasure today to introduce Ranu Jung. She's the Wallace H. Coulter Eminent Scholars Chair and the new Chair of Biomedical Engineering at Florida International University. Ranu's twice an case grad, doing her MS and PhD when she came here, working with Peter Katona. She spent about 6 or 7 years at the University of Kentucky, where she was a tenured professor before moving to Arizona State, which a number of us in this room know well. And before she just last month, I think, went to Florida?
About a month ago.
A month ago, so she's still in flux. So she has quite a long record of accomplishments. When she was at Arizona State, she founded the Center for Adaptive Neural Systems. She's the founder of the Organization for Computational Neuroscience. She's had numerous companies and lots of translational work or projects, so I think that goes well for the type of flavor of speakers we've had here. I think today she'll be talking about some of her recent work using computational models, neuromimetic systems to design neural prosthetics and design new systems. So we look forward to your talk, Ranu.
Thanks. Thank you so much, Ken. This is wonderful coming back. And when I got into the plane yesterday morning, they said 3 degrees. And they said it's warmed up by the time we were landing, and it was 14 degrees. I was like, okay, how did I choose to go there in February? But, but I was taking pictures of the snow. Anyway, so I'm going to give you a little bit more than the recent work, kind of try to give you a little bit of a history of where we're coming from. The blue colors are my new colors. I'm at FIU now. But since I've been there only for about less than a month, a lot of the work that I'm presenting to you is really coming out of the work that I had done at Arizona State University as part of the Center for Adaptive Neural Systems. So this is the word that I'd like you to keep thinking about is neurodesign. So the concept here is that what is happening with the nervous system is activation of patterns, and that these patterns that are being activated are creating function, are creating output, and are affecting the nervous system itself at all different scales. In any case, to get information about these patterns, you're basically accessing that information, and that's what you would call investigation, or the basic scientific inquiry about what is going on with the nervous system itself. The other thing is being able to influence the system, so influence these patterns of activity. And this is usually the term that we would often use here would be the neuroprosthesis thing, where you are replacing lost function, maybe because of deterioration or some trauma or something that has gone wrong with the nervous system itself. But then the third part of it is being able to repair the system itself. So in this case, you are effectively promoting learning and learning at many different scales. And again, what you're trying to do is alter the overall pattern of activation at multiple levels. And so these 3 words, investigate, replace, repair, are going to be part of the theme with which we have been trying to work under this broader picture. So when we talk about neurotechnology, then the technology is basically interfacing with the nervous system. And when it is interfacing with the nervous system, it's at multiple scales. And the scale can go all the way from molecular, cellular, I didn't put the genes and the proteins and all of that, and with more and more complexity. And ultimately, especially being in the engineering field, we are looking at applications. And obviously, if you're talking about medical applications, there's a societal impact. So there's this interface of technology that is occurring at all different levels. And the whole idea being investigate, replace, or repair. And as we are doing this technology interface, one of the things we are trying to capture is, can we learn something from the biology itself and do biomimicry in our design of our technology? And why do that is the idea is that you are going to perhaps make more robust and versatile systems if you can capture the versatility and the robustness of biological systems itself. And then, of course, because you're interfacing, then that means that you are integrating with the nervous system or the musculoskeletal system, that any way you combine the two together. And if you are looking at causing repair, which means promoting changes in some way, then the whole hope is that this integration that is occurring is effectively an adaptive integration, so the technology can be adaptive as you are promoting the adaptation in the living system itself. So this gives you the bigger framework under which different things we have been working on fall under. And the three pillars are, which the pillars are getting lost in this. There is right there is neuro rehab, there's neuroscience, and there's neural engineering. So that's all put together. So let's hope that we don't have too many cutout slides there. Then, of course, you often hear of bench to bedside. But one of the things that I want to point out is a lot of the work is coming from bedside to the bench, back to the bedside. And the pipeline of translation, and you could belong to any single box in here. You could jump in and out of here from wherever you want to. But the whole point is to take ideas and discoveries all the way out to assistive and therapeutic systems and delivery. I wonder if I need to change the font present. OK, there are different things that were going on and application areas in the center. The two areas that I've been primarily involved in have been with regards to spinal cord injury and limb trauma. And so what I'm going to do today is to give you some vignettes, maybe of some examples of things that I've done over the years that fall effectively under these two areas. At the center, we had the capability, or we still have the capability of doing a lot of these human subject studies. And a similar setup is now what I'm setting up at Florida International University to be able to do the biomechanics, to be able to do the cardiovascular and respiratory measurements, but also to be able to do some non-invasive brain imaging capabilities. That's being set up there. Lab animal studies, as some of you know, I started out here doing in the dog labs up in Wickenren and doing work with those. So I have worked across different animal species during my own research career. And so those at multiple scales. And so those animal studies are going to continue, and the labs are being set up. And of course, modeling and computation go along. They are the core that are there for all of these studies. So if I look at all the different research projects or different things, you could kind of group them into three focus areas. One of them is what we would call neuromorphic control systems. And so here you have got control systems inspired by biology. They're not necessarily exactly looking like a neuron or something like that, but it could be. But they're inspired by the biological system. The second area is applying these neuroprostheses and advanced prosthetic systems. And this is the technology to replace lost or missing functionality. And the third is the area of adaptation in neural systems, so to promote learning in the nervous system. So I'm going to pick an example from each one of these and go through all of them. So let's starting with the control system. So if I look at this over the years, now this spans quite a bit of time. There have been steps that I have gone through personally over the years, and sometimes you don't know where you might end with what you start. You start with something and it ends up somewhere else. So this is work that I started with. Who knows what this is? Lamprey. So for the students who may not know about it, it's a jawless. It's an agnathan. Pardon?
It's a bureaucrat.
Bureaucrat. Yes, that too. Toothless. It has a spine, though. So the studies there. So that's the lamprey. We're going to talk about the lamprey, not the trout it's feeding on, and then look at how investigation in there has led to some new spinal interface work. And then ultimately has that inspired into actually application for powered limb splints for a particular application for the injured soldier. So here's the lamprey, and in this case, an excitatory amino acid was injected into the gut of the animal, and when you inject this into the gut, the neurotransmitters are activated in the spinal cord, and you actually get this rhythmic pattern. But you can see if you record EMGs, it's not quite doing very well. It's got some rhythmic activity, silence, rhythmic activity, silence. So there are loops between the brain and the spinal cord that are getting activated, and there's a conflict between descending inputs and ascending inputs, because here we have artificially excited the spinal cord. Same lamprey, you transect the spinal cord, and here you can see that it's swimming very nicely. So there's two things that are happening here. One is that there is this so-called pattern generator or central pattern generator in the spinal cord itself that is getting activated pharmacologically, and that is allowing this rhythmic movement to happen. You have cut this closed loop pattern, the feedback feed-forward pathway between the brain and the spinal cord, and you get this beautiful activation. So a side story, but if you have incomplete lesions and you start to do pharmacological activations or spinal activation at the spinal cord level, and you had these loops happening with direct volitional control and feedback that you have artificially created, what would that mean for stable, robust movement patterns? Something to think about. So in any case, a lot of different studies that they conducted were to look at these brain spinal cord loops and doing modeling. And then what the nice thing is you can take the brain and spinal cord out, you can stick it in a bath, and after you stick it in a bath, you can record from ventral roots, you can record intracellularly, you can stimulate. So you basically have a system in a dish that you can now probe quite a bit. And based on that, what we know and what several other people, especially Sten Grillner, who did a lot of work with this as the model system for looking at motor control. From multiple people's research efforts, you can come up with populations of neurons, this E excitatory, cross inhibitory, and lateral inhibitory, and this core basically forms that central pattern generator. And this, in the case of a lamprey, you can conceptually think of it like a chain of unit pattern generators where there is an individual unit pattern generator. They're not separated like in a leech. The connectivity is there, but effectively it functions like a chain, and you can take out up to 2 segments and stick it into a bath and look at what happens. So one of the things that we then when we were looking at this investigation, and I was looking at the brain spinal cord things, one of the things we wanted to look at was could we create an electronic artificial spinal segment? And if we did, could we then interface it with the rest of the chain and be able to do a closed loop? What would happen effectively if we did a closed loop control? So this was, as far as I know, one of the first times that anybody had done a spinal interface and a spinal stem. So I actually collaborated with another colleague, Liz Brower, who is an electrical engineer. And we ended up putting together an analog VLSI chip and some discrete components together to come up with a single segment of the spinal cord, basically the model that was there. And then this segment can give you the rhythmic pattern that you just saw before. And now the question was, can we interface it with the spinal cord and get a whole chain together? So in this particular case, so the idea is you have this chip, this electronic segment, use that to stimulate the lamprey spinal cord. Or in the bath, you can activate the spinal circuitry of the spinal cord and get this fictive locomotion, because there's no real muscle there, only the spinal cord is there, and it gives you the rhythmic movement pattern, and use that to drive the chip. And could you get a closed loop working together? So here's the picture. So here's the rhythmic output from the electronic UPG, or unit pattern generator. Here's activity in the induced by N-methyl L-aspartate or excitatory amino acid in the spinal cord. Some activity is there. Here you start the stimulation, and as soon as you start doing the stimulation based on the output of here, you get a phase-locked pattern in the spinal activity to this electronic cyclic input. But what you'll notice is that slowly this entrainment is lost after some time. And one thing else that you'll notice is that you can kind of go and you can make this rhythm go faster. You can't make it go slower. You are basically exciting the system. Now, this could have been done effectively with any kind of a stimulator. And you could have done that just with the stimulator itself. Here, what we are looking at is you have got output from the spinal cord now, and you are inputting that into this electronic UPG. the thing that you have to notice here is that it's not just a matter of changing the frequency of this unit pattern generator, but you effectively change within the cycle, you are changing the whole morphology of the pattern itself. So it's a nonlinear process that's going on, and you can change what is going on in a very tiny time scale, and the whole morphology changes. Now, when you put the two together, then what we noticed is that you can effectively make the circuitry and the spinal cord lock together. And when you can lock together and you can actually make it go it can go slower or it can go faster. And it is these nonlinear, so it's one more in the chain of the pattern generators that we have now created. But since we have this electronic circuit, we can then entrain this electronic circuit with another input. And so we can make this go faster or slower, and then the locked system can go faster and slower. So this was a proof of concept of being able to say we've got a nonlinear electronic UPG pattern. The design is based on what we had from the biological system, and we can put it in and interface it with the system. It's not just a matter of making it go faster or slower, but can entrain it to different frequencies. So fast forward to a few years later, there was a request from the Army for neuromorphic control of powered limb splints. And what they wanted was that somehow get wounded soldiers out of the battlefield. This was, the Iraq War, starting with that. And they wanted they want you to get this person by themselves, walk out of their place, and be able to use some kind of a powered limb splint story. So in their announcement was a little reference at the bottom, and the reference was to my paper on the lamprey with the spinal interface. And so I was like, but there was a little catch. You needed a company, and you couldn't, it wasn't something you could do from the university side. So, so guess what we did? I formed a company, and we then bid for this for this project. And as it turns out, we got the project, and we ended up using some of the approaches that we had done with the lamprey work to now design a powered limb splint. And so everything that was there at that time, there was some early work from Hugh Hurst's group at MIT, where they had started to make an active ankle-foot orthosis, but primarily everything else was a passive orthosis that was there. So the concept that we came up with, because you had to be able to get somebody with the human intent should be there, and that powered limb splint should be able to move, get them out of there. It must provide stabilization to the injured lower limb. It should, because that's what is happening to the soldier. You couldn't custom design things. You had to do something that would fit a large guy or a small guy. You have to be able to drop it off, into the field. So all these multiple constraints. It has to be battery powered. It has to be tetherless. All things that if you could design it, you would not really do all of those things for a clinical application. But anyway, the idea that we came up with was effectively a false foot. So you've got the leg braced in there, and then you have got a false foot here, and this false foot then is the one that moves up and down, and that's what is actuated, and it's controlled, and then you wear this boot and you basically get out of there. And so initially when we started out, we had it being triggered to get the person's intent from sensors on the opposite leg, but ultimately because you couldn't say, the person will be able to put sensors on them and take them off, everything had to be on this particular device so that the soldier would just wear that and get out of there. We started with, again, using some of the pattern generator concepts that we had done before, because this is effectively a rhythmic pattern that is you are getting. And with large breadboarding of all of that circuitry, we ended up doing another VLSI chip design, putting it into a smaller board, and then another whole smaller board, and figuring out where should the sensors be, what kind of sensors should be there. Person should be able to walk on step over things, should be able to crawl, should be able to jump, and you have to be able to capture information from them. We ran into some problems with the motor and the analog input thing, so we ended up having to put some stuff in a backpack for the initial part. But in any case, we have a tetherless system that a person can wear and can walk with that. So in this case, what is happening is that the one leg is stabilized. It's not the foot, ankle is stabilized, and the false foot is moving, and then the person is basically walking around with that. So we've filed for patent for it, and let's see where it goes. But if we could miniaturize this, and if we could get the weight thing under control, and if we didn't have these constraints that you must walk for so far for so many hours, then effectively you could get crutch-free walking. That is the concept that it would be to go into everyday use. So that's one of the examples of going through a neuromorphic control system story of how that has gone through. The next thing I want to talk to you about is an adaptation in neural systems. And I'm going to switch gears to talk about spinal cord injury. I don't need to tell this audience anything about spinal cord injury, but the spinal cord is not a conduit, as you all know. And we just talked even about the CPG structure, and it has smarts of its own, And I also don't need to tell you what all happens with spinal cord after spinal cord injury. But anyway, there's a multiple primary and secondary pathophysiological changes that occur, loss of function because of disruption, and there are many, many different experimental approaches to give you, to prevent the deterioration after injury, the protection part, and then there are people trying to stimulate some kind of regeneration. And then if you have incomplete injury, you have this whole concept of trying to promote plasticity or within the spared nervous system itself. And again, right here, within the centers here, you are doing several of these things. And and I'm going to tell you some of the work that came right out of here that is still carrying on with me. At the very bottom, what you see is work primarily done by Jimmy Abbas. And many of you guys know Jimmy. And so this is incomplete spinal cord injury person. And then there's a handheld system that Custom Kinetics, a company that he started, is now something that you can take home and be able to do surface stim and be able to provide rhythmic movement pattern. But the question that we were asking is, what happens when you do this repetitive movement therapy? What is it that what mechanisms are there that are getting affected? How are you going to bring about change in the nervous system itself? And so you can, of course, look at people to whatever extent you can, but if we had an animal model, it would open us up a lot more options for us. And so the animal model that we decided that I would work on was the rat. And part of it is there's a lot of basic science studies that are going on with the rat model and now the mouse model for just looking at spinal cord injury itself. So there's different things that we are doing, many different aspects of it. We are looking at investigating function, changes in the function itself. We are looking at the circuitry in the spinal cord itself, what happens to that. And then we are also looking at morphology of what is happening to the neurons, et cetera. And then, of course, the goal is that you are interfacing and providing the therapy, and how would you affect all of these. I'm just going to give you very briefly a little bit of information here with regards to this particular aspect about motor neuron control. One of the things that happens after spinal cord injury is this persistent inward currents that are there in the motor neurons. Those are enhanced, and they are thought to contribute a lot to spasticity, or the self-sustained firing. So you put an injected current in, and when it comes out, rather than shutting off after chronic injury, the neuron keeps firing. And so that motor neuron output would affect the muscle contractions and cause spasticity. Other aspects that one has to look at is also what is happening to the inhibitory circuit. And GABA and glycine have been thought of as being important for this inhibitory control of the circuit. And then there's also intrinsic properties like depolarization shifts, et cetera. So in this particular case, we have been looking at some computational modeling looking at models of the neurons, small circuits, and examining what happens to the behavior of these as you change change different aspects of either changing the persistent inward currents or changing these inhibitory connectivity. And you can do all kinds of bifurcation analyses. You can get the same kind of patterns that you get in the actual experiments. And one of the things that has come out, which again, this is falling out, is that if you have no inhibition, and if that would be an impaired inhibition, you get this kind of a hysteresis pattern that would be there. If you had inhibition of both GABA and glycine, you would lose this hysteretic pattern. But if you only remove GABA, then you don't take this out. And if you remove just glycine, then you get this no inhibition. So basically saying that GABA is the target that might be the one that you can control. So that's a new target that one could think about with regards to being able to control specifically for another target that you could think about for controlling for management of spasticity. And the question is, can we come up with neurotechnology that could somehow target inhibitory circuits? I don't know, but that would be things that one would have to think about. But one of the things that did fall out of this is that it's not glycine, it's GABA, even though right now both of them are being thought of. And then we've also looked at them depolarization shifts that could occur after injury that can be targeted by molecular targets. But with this GABA control, we can actually overcome a lot of those problems that happen just with the molecular shift also. OK, so then that was a little bit here. I'm not going to talk anything about this. And then I'm going to talk a little bit about this now about the gait control part. So there's a rodent model that we've been working with, incomplete spinal cord injury. A lot of it right now is doing measures of outcome measures. Those of you who work with people, you have the ASIA scales. There is an equivalent BBB scoring scale for rats that you see how they walk, what happens to them. There used to be very little kinematics done. So we were first ones who did a whole 3D kinematics for the entire rat gait to figure out what are the actual patterns of movement. And we can also do like a 2D analysis of the movement pattern. But here's our little rat walking there, and you put the markers, and you've got cameras on all sides. And then you can actually look at the patterns that you get. And you have implanted EMGs, and you get, so these people those of you who actually do human kinematics are quite familiar with these and EMGs. So the novelty here was that nobody had looked at this in a rat model. And so we have got this now basic work for saying what happens with the rat model. So you've got the hip angles, you've got the EMG patterns, the knee pattern, the ankle, shoulders, elbows. And then you get, I just want you to pay attention to this figure in particular, because I'm going to bring this up later. So this is the left and the right knee pattern. So it is basically the 180 degrees out of phase and kind of a figure 8 and a butterfly kind of pattern that you get. This is the normal case. So now this is a rat that is a normal rat and walking on the treadmill, 2 weeks post-injury, and it's dragging itself, but it's incomplete injury. If you give it support, it does a lot better. So a lot of the problem is posture, as many of you guys know, and not necessarily anything to do with the pattern generators, that it's the posture itself. And then this is spontaneous recovery at 13 weeks post-injury, and it has recovered quite a bit of the function. If you look at the kinematics, you will still see that there are problems. So if you just let the animal recover by itself, this is at 4 weeks. This is a moderate level of injury. This is the pattern before, and this is group data from multiple animals. And then here what you see is you kind of get a stiff gait. Like a stiff gait kind of pattern that shows up after the 4 weeks. Okay, so keep that picture in mind. This is just normal recovery of the animal. Now if we look at, we said, what about giving repetitive movement therapy? So there's 2 ways we could do it, using neuromuscular electrical stimulation or the traditional treadmill training therapy. So to do the neuromuscular electrical stimulation, we looked at what did Case do with the intramuscular electrodes. So we said, OK, we will use that approach. It's a lot harder to implant tiny fine motor points for little rat muscles and do the implants. But so we spent quite a bit of time doing that, and we actually ultimately have worked all of that out to be able to do the stimulation for multiple muscles and be able to get movement patterns for that. And we did what stimulation frequencies you should use. You have to go at much higher frequencies of stimulation than in humans because there's mostly fast fiber in the rats versus in humans. But we did all of that. And then we did a study where we said this is now just open loop stimulation, and it is just hip flexors and extensors that are being stimulated on a regular basis. Animals have been implanted, and after being implanted, there's injury, 7 days pre-stimulation, and then for 1 week, effectively 5 days of stimulation is all we have done. Okay? And all the pattern is doing is left, right, and it's just open loop stim, no closed loop part. At the end of it, what we do is we say, okay, walk on the treadmill. We look at the kinematics, we look at the patterns, we look at the footfall coordination to see interphase relationships and see how much recovery has occurred or not. And here's the story then. So this is intact animal group. Here is the group that had no therapy and a mess. And here is the group that got the 1 week of therapy, or less than 1 week, 5 days of therapy. And you see that the same stiff-legged pattern has come back. This is this is just with that 1 week with open loop stim. Okay? So now if I look at treadmill therapy, we looked at a greater level of injury, and we are doing treadmill therapy, but we took the animals for 4 weeks of daily therapy pattern, and then we look at the treadmill patterns again and see what happens. So here's now the whole story put together. No therapy, 2 weeks post-injury with a moderate level of injury, you have a mess. Neuromuscular electrical stimulation, you get the stiff-gated response back. 4 weeks of recovery, no therapy, you also see a stiff-gated kind of pattern back. So this looks similar to this. This has just occurred earlier. If you look at a higher level of injury, you can go up to 6 weeks, and there is still not a good pattern without any therapy. And if we did treadmill therapy, only 2 out of 8 animals gave us anything. And these are starting to get something back, but only 2 out of 8 gave us some kind of a recovery back. So what it suggests is that perhaps there is a window of opportunity to apply electrical stimulation maybe early enough after injury, where you might be able to accelerate the recovery of what is happening. Why would this be? Remember, all we did was left, right things. The good part would be that you would not have to put people up into their, into a treadmill, into a harness. You could keep them lying down and still do some of that therapy. And perhaps all it would do is prime them for greater, treadmill therapy or something else later down the road. The other thing then we said is, well, we've only been using this pattern that is a open loop kind of pattern. What if we actually did a proper pattern and did a longer stimulation therapy for several cycles, so that, because as you all know, with the who work with functional electrical stimulation, that you stimulate the muscles fatigue. And so you are not able to actually get movement pattern for a long time. So can we do that? So what we did is, for this, is I went back to Jimmy's pattern generator, pattern shaper, adaptive controller. And so this is perhaps, Ron, this might actually be data from when he collected. Well, maybe not. This is somebody's legs in Kentucky, so I know that. But if you do the stimulation and you look at how, when you do the stimulation levels and you look how much angle you are getting from, in this case, this is just the at the quad. So this is just looking at the angle over here. So you get a nonlinear pattern that is there. So you have got a nonlinearity. You can have a steep slope. And then if you do the stimulation for some time and then you repeat it, then you basically, there's a change in the slope itself. So effectively saying muscles have fatigue. So you have to deal with this different kinds of muscles for different people, nonlinearities in them, and then you have to deal with fatigue as you continue to do the stimulation. So Jimmy had come up with an approach which borrowed from the biomimetic idea of what is going on with the brain, with the neural control in the spinal cord itself. And in this case, if we look at what I talked to you before about with the lampreys was the pattern generator part. You can think of the pattern generator part going out and the output going to all the motor neurons, and the motor neurons basically act like filters to take this rhythmic input and shape it to give input to different muscle groups. And so then you have this concept of a pattern generator, pattern shaper, where any rhythmic output is being somehow managed to go into a certain pattern to stimulate the different muscles. And so that's the picture that is there. But now what you need is you need this pattern shaper to adapt online. So it has to keep changing as the muscles fatigue or as the neural activity is changing so that you can get the desired outcome that is there. And so you can customize the stimulation, you can account for nonlinearities in dynamics, and you can adjust for fatigue. And the pattern shaper, you have to look up Abbas's papers, but it's a single layer neural network. It's got, each of these neurons has got an individual pattern shaper neuron and neuron activity. Output is like basis functions that are there that are spread out in time. And at any given time, multiple of these will add up to give you the peak activity for that muscle output. And how much you get as the output going there depends on the previous outputs, and there's a learning algorithm that goes on along with that. So we took that. He had done it primarily for single joints. He's also done some stuff with 2 joints, but they were not flexion and extension at the same time. They're using peroneal stim to get the flexion part. In the case of the rat now, we've got both the flexion and the extension, and then we needed the left and right aspects. So now we put this together, and to be able to do it in the rodent, we had to change what we had to do with the updates, et cetera. But anyway, that was the system. So now you've got the rat on a platform, cutaneous input, because that is suggested that helps also with the reflexes and may help with the recovery. And you've got, it's much harder to make little potentiometers and goniometers for rats that you have to put on top. But anyway, we can do the measurements of that. You get the outputs. It goes to these flexes and extensors. There's the adaptive controller. And we put that into a loop. There's our subject. And there's the rest of the stuff that needs to go with it. So here's what's happening. So this is open loop control. So our desired pattern is this rhythmic oscillation. And this pattern is what we got from the kinematics of the hip kinematics as the rat was walking on the treadmill. And then if you just did open loop stim, you would get flexion extension. So if you didn't try to tweak that open loop stim, you would just basically get this. After some time, initially you get this bang bang movement. So this is the angle of the hip angle you are seeing, and internal angle. And then after some time, it is basically the angle is reduced because range of motion is reduced because the muscles have fatigued. In the bottom here, what you're seeing is that adaptive controller. You start out with nothing. You are not tweaking there, sitting there and saying, I'm going to tweak this value or tweak that value. There's online learning, and you can see within a few cycles, you start seeing this is the stimulation output currents, and you start seeing what is happening, and you can continue to have this pattern going on. So these are initially were in the anesthetized prep, and then we did it in the chronic prep. We implanted them. And then the question was that we really wanted to go for long numbers of cycles. So we came up with an intermittent stimulation paradigm where we do 5 cycles. There's a little bit off time, 5 cycles, a little bit off time. But we can go, that's all we tested is for like over 500 cycles that you can continue to go. And once you can, the error remains low. Look at different muscles, different number of animals. And what you see is that the error will remain low. In some cases, depending on the angle sensor position, there are some, because of that, there may be an error. But so you may have to give more current or put more charge in. But basically, there is no real pattern that falls out of that. You get very good tracking, and you can keep this going. So now we do have a model in which we could do adaptive control. So the next step, this just finished last year. And as soon as I can get the whole lab set up and move things over at FIU, then we can now start to think about, should we put this adaptive controller algorithm, and would that help us get better recovery and not just early recovery with the electrical stimulation? Okay? And so the good part for us is that in the human subjects, we already actually have the capability of doing surface stim. And so there is this translation that can happen between an animal model and the human model. And it actually, as I said, came from the bedside to the bench to the bedside, not the bench to the bedside part. The last part that I'm going to talk to you about is this neural prosthesis and advanced prosthetic systems. And many of you here are working on many of these aspects, and especially last week's speaker, Dr. Durand, has been working with the fine electrodes things. So you are familiar with this whole aspect of wanting to give sensory feedback to people who are using prostheses, and then doing bidirectional communication to be able to get both motor control and feedback. And of course, the scientific question to me that is of a lot of interest is how do you come up with co-adaptive learning or synergistic learning between these engineered systems and the nervous system itself, as you would use these prostheses that give you sensory feedback at the same time that you do the motor control. We are nowhere close to doing this. We would like to be the postage stamp that can get to doing that. We have started work on some of this part. And so you are all pretty aware of all the different kinds of electrodes and arrays that are being used to tap into the neural activity to be able to control the prostheses that different people are using, different groups are using. The work that we have been following is the approach that was taken by Ken Horch. And Ken Horch is a member of he was a member of the Center for Adaptive Neural Systems, moved to Arizona. He's now faculty at FIU. So so he's an integral part of our team to do that. And so the whole, of course, the idea is that you'll get sensory feedback may allow reduced need for visual attention, improved dexterity, improved body image. And then the long-term amputees, they do have partial atrophy and degeneration. But one of the things, and they have changes in cortical maps, but one of the really interesting things that came out of these papers from Ken, Dylan and Ken Dylan and Horch, 2004, 2005, et cetera, is that you have the sensory and motor capabilities in long-term amputees 30 years later. They had subjects who had about 10 months to 30 years later, and you could get activity from you could do both sensory input to them and get motor control out of them. And that was the most exciting part to me when I think about it, that what is this happening with the whole plasticity issue that is going on. So anyway, the opportunity here was to come up with direct affer-neural sensory feedback. And the electrodes, for those students who may or may not know about this, are what I call they are intravesicular electrodes, and they are longitudinal intravesicular electrodes. So they're going along the length of the nerve. In the fascicles, there's a bare spot of about 1 millimeter that is exposed. The fascicles are actually exposed when these are threaded in. So it's kind of threading a thread through with a needle that is put through, and then that exposed part remains inside. So it's basically local stimulation to wherever you have put that where part is, and it is a very small amount of charge that you need to put in because you are right inside the fascicles itself. And so the original work, there's Motion Control is the company with the prosthetic hand that we are working with. And Cochlear Limited is our another device partner for us. Artificial Limb Specialists are prosthetics group. And the original group had Mayo Clinic with us at FIU. Now we are the surgical team is changing for that. So this is data from Ken. And so what he had done with his studies was they're percutaneously placed electrodes, and these electrodes were placed in. And what you're seeing over here is a plot that is showing where, and I'm going to show you a video of some of this, that you basically have a subject, and the subject has been told that the one intact arm is put at some position randomly, and then the subject is blindfolded, and that subject needs to think about moving their phantom arm to that particular position. And what they find is that they have got a pretty good match of what the subjects are able to do with their, with thinking about where they're going to end up being. So this is a small video. And so in this particular video, you have got these percutaneously implanted electrodes in the stump, in the upper arm, and the subject is blindfolded, and this prosthetic arm is going to move, and that movement is controlled basically by the output from what is being recorded from the stump, but the subject is going to indicate what he is doing by his intact arm. And so when you see that, so you can see that you get it's a fantastically well-regulated capability of doing this control. But of course, this is a single, it's not multiple electrodes, it's not multiple, they're recording for one single thing, one single joint. There's no sensory feedback going on at this time. So there are lots of limitations that are there for that. And it is all, it has to do with being able to get percutaneous leads out. The other part that he did was with the sensation, and this is very, very interesting too, because what he has shown is that you can get discrete sensations and graded sensations. And in this particular case, the subjects are now being stimulated, and depending on the amplitude of the stimulation, you can get either, so if the subject may say, I feel the distal tip, and if you increase the amplitude, then either the stimulation goes proximal in, they perceive something going proximal in, or it jumps to another fingertip. So but you can get fairly discrete sensations. The other thing is that if you do change in frequency of the stimulation, then you can get a graded control of what is happening. So in this case, they were indenting a force sensor on a prosthetic hand, and the subject could say, oh, I feel it more. And depending on more the indentation was, or with regard to an angle on a prosthetic elbow, they could say, okay, this is what is happening with it. So what we are doing now is, based on this, is basically we are working on now taking it to chronic subjects, but also taking it to be able to do a fully implanted system where we can do a wireless communication. So it's basically a wireless communication inside between what is implanted in and what we are doing with the ability to do the sensory part. So it's the loop that everybody knows about, but in our case, the control of the prosthesis right now this is for human subjects, is just going to be the traditional motion control, EMG control, so we are not working on getting that part to be anything more sophisticated there. And then you have the prosthetic hand, however, got sensors on it for both for proprioception, opening and closing, and for touch, and that information, we're doing some custom hardware, some custom software, and then wireless peripheral nerve sensory stimulation, and closing this particular loop. So the technology, developing this technology for that. At the same time, we are doing technology for recording, but this is only right now in the animal stage, and this happens to be an intravesicular recording from a rat sciatic nerve, where we have basically moved the foot back and forth, up and down. So that's the project that is consuming a lot of time, and we've been working on that. And Jimmy is, somebody was asking me what's Jimmy doing. Is a lot of work on this that he's been working on, has been on this part of this project also. So idea then, ultimately then really what we would like, if we look at the whole thing together, is to come up with this whole aspect, to try to do adaptive neural systems. So neurotechnology that is increasingly integrated with the biological systems, neurotechnology that perhaps could be made more and more adaptive in its capabilities, neurotechnology that could provide targeted sensing and activation, and then effectively neurotechnology can be personalized. If there is something called personalized medicine, this neurotech interface is personalized to you, for you, and locally, and has a local control thing. Of course, the key issues are, how are you going to integrate adaptive systems which are on both sides adaptive with each other. That's examples that I've given you. None of this work is possible by one person. It's a huge team that works on it. And more recently, the work has been with Jimmy, Sharon Crook, Ken Horch, Steve Phillips from electrical engineering, Marco, Tom Ham from BNI. I didn't show you some work from him. He has been a fantastic neurophysiologist. We just published 2 papers where he was able to do intracellular voltage clamps from rat neurons, which is really, really hard to do. So that we are very excited about it, and we see the changes that are happening after injury and in normal animals. Victoria Booth is a mathematician. Liz Brower, who did some of the original VLSI work. Different members of the staff who have worked with it. The small company that I had, engineers with there. Cochlear, motion control people, Mayo, artificial limb specialists, bunch of postdoc fellows who have worked for a long time. Kazuhiko and Tsukasa were 2 MD, PhD orthopods from Japan who really, without their help, we would not have gotten the rat model moved as fast as we were able to, because it was amazingly time-consuming and hard to get done, and several graduate students. Can't thank enough the funding agencies without whom the work could not have happened. 4 different NIH institutes have funded the work: NSF, Army, and Science Foundation Arizona. And then here are pictures of my new abode in Florida International University. I'll take questions.
All right, well, thank you very much, Ranu. We have a small token of our appreciation.
Oh, thank you so much.
It's not a space heater, but it'll help out. So we'll open up for questions. I'd like to ask you, please speak into the microphone so we can capture everything. So we'll open it up now.
Thanks, Ranu. That was great. I was curious about the study talked about where you had the stimulation versus the treadmill training. Mm-hmm. And I want to make sure I understand this correctly, and you can comment on it. You intervened at the hip. You stimulated the hip flexors and extensors.
Yes.
Your outcome measures were at the knee.
No, so, I highlighted the outcome measure for the knee, but if you look at it, the outcome measures are the whole gait. So right. The coordination pattern of the forelimbs, hind limbs is improved. The symmetries at the hip or the knee are improved. So all of those are improved. Ankle is not, it does not get affected much.
Okay, so can you comment on intervening proximally and their effects on the distal kinematics?
So we have not done anything directly with regards to just doing the knee stimulation or doing the ankle alone. If we did the knee alone, you would have some of the effects will happen on the hip also, unless you stabilize the hip and keep it there. Ankle can be kind of kept completely separately from what is happening to the knee and the hip. So I'm not sure necessarily with the ankle how much we will or will not get. And there's a lot of drag that occurs with the ankle. The other part for the ankle is that in the incomplete injury part, even though you may have like co-contractions of the muscles, you can still they can still overcome that drag, and it's dragging, but they can pick it up after some time. And this is a study that I didn't talk about, which one of the doctoral students looking at, is what happens? How come you can do this? You still have these co-contractions happening, and still you can do this lift off, so to speak. And what we found is that if you look at the overall shape of activation, so if it's like a squared shape of the EMG envelope, right? If that's the case, if that kind of that shape changes to be like a sloped region, so by changing that overall pooled activity, you can overcome even co-contraction. This is in simulation, in co-contraction to give you a lift off. It will still not be completely normal, but you'll be able to get that off. It's in simulation that we are seeing that. So, so going back to what you're saying, just doing the ankle, would it affect the rest? Not in this model, I don't think so. Knee, perhaps I think we might be able to get it back up. Yes.
Hi. Do you think that the improvement that you had in the rats with their walking was a reflection of recovery of connections from cortical centers to the spinal cord, or improved function of the spinal cord generators being able to operate better after time and with practice?
It's not an answer that I can easily directly give, but it's a very short time. And so in that short time, how much cortical spinal pathways have changed is hard to say, even though Schwab's work says you can get very quick plasticity occurring there. But is there really any sprouting, any regeneration like that? I don't know. With regards to the pattern generator part, what we do know is that if we do electrical stim, there's two things that is happening, right? One is that if you do rhythmic electrical stim, you're contracting the muscles, you're getting sensory feedback that was missing, that is still going back. The other thing is that even if you don't get contraction, Tessa Gordon's work and all have shown that you have got upregulation of neurotrophic factors in the muscle itself. So the electrical stim itself is doing that, and those neurotrophic factors are getting retrogradely transported to the spinal cord. And then there's a whole group of people that have been looking at like upregulation of BDNF and things like that in the spinal cord and arguing for that having an effect. We really don't know. We also know that from a paper in TINS where they looked at some of the changes in the genomic profile of what is happening soon after injury, that there are certain receptors upregulated or downregulated within 1 week. So could it, and some of them are neurotrophic factors that might help or promote recovery. Could it be that that is a magic window in which you can influence that? I don't know. So it's not regeneration of axons for sure. Is there some other, other neurotransmitter, neuromodulators that are being affected? Can't say that.
Just one question. Where is Ken Horst taking his life technology? Because Ken Yoshida has done all his work with polyimide. Is he going in that same direction?
Well, Ken Horst is now with us. And he is doing some work still at Utah with some implants that are still going on. None of these are with the polyimide electrodes right now. So all of the technology, as of now, right now, is with the wires, yes. But does it mean that it will not happen or cannot happen? Yes, it can happen, but currently it is with this particular grant, it is with the wires. And then the Utah work that he's also doing right now is with the wires.
In the prosthetics experiment, did you do a control in which the subject actually intended to lift the intact arm and prove that the prosthetic arm did not follow that?
So those are studies from Ken from before. So those are not, the subject is not connected directly to the prosthetic arm anyway. The prosthetic arm is sitting aside. So it's not something that they are lifting and picking it up by themselves.
Yeah, if they think they are lifting with their injured arm, the prosthetic arm.
Yes, it will.
So if they actually think they are not lifting the injured arm, but the actual arm, would the prosthetic arm follow that?
Well, they're not. The only way the prosthetic arm is controlled is because of the recordings from the amputated arm, not from the normal arm. So, anybody else?
Anyone else? All right. Well, thank you very much.
Thank you.